Thank you, Dan and Andy and Brian and the Greater Yellowstone Coordinating Committee. As mentioned, I'm Mike Schwartz. I'm the Wildlife Program Manager for the Rocky Mountain Research Station. And today I'm going to talk about connectivity and mesocarnivores in the Greater Yellowstone ecosystem. When we think of carnivores, we think of rare carnivores, we think of these two species all the time. If I asked you, what are the two carnivore species that you think about in the Yellowstone ecosystem? 95% of you are going to think of these two, right? But that's the wrong way to look at the problem. When we put abundance on one axis and we list wolves and bears and wolverine and lynx and fisher on the other, we see really quickly that the real rare species in the ecosystem are the wolverine, the lynx, and the fisher. We have no good population estimates for these, but they are probably only 10 to 20% of the size of these other things that you would instantly think about when you think about rare carnivores. So I really appreciate being invited today to talk about the real rare carnivores of this ecosystem. The only trouble that I have is that this is a migration symposium. Now, mesocarnivores don't migrate as far as we know. We have no stories of wolverine thundering across the tundra, right? They don't migrate. Okay, so take this home. Now I know this is the problem. I put that up there, this cute little video from Russia that's a weird anomaly, and that's the only thing you are going to remember of my entire talk. But try to skip over that memory and go to this memory. Mesocarnivores don't migrate, but they do move. And this ecosystem is important for their movement. That's the take-home message. So for those of you that want to now go to sleep, you can. I'll walk you through to get to that point again at the very end, that this is an important ecosystem for mesocarnivore movement. And I want to be really clear what I'm talking about with movement, because I'm a geneticist and we're kind of weird. We talk about movement in a very different way than a lot of other folks. So I want to be clear that we have migration. This would be movement from one place to another to gather resources or to optimize resources, or as I learned this morning from Arthur's great talk, to potentially avoid predators. And as biologists, we quantify that by looking at pathways and flyways and migration routes. That's one type of movement. We have this other type of movement, this territorial movement. How else do you represent that, right? You have territorial movement, and we oftentimes quantify that by looking at home ranges, and that is movement to gather resources in an area, often to defend yourself against conspecifics. And you see many maps like this one from Algonquin Park that looks at territorial boundaries of different wolves in an area. A third type of movement that we may talk about is exploratory movement. And this is a mountain lion that was killed in Connecticut. It was a few years ago. It wandered all the way out from the South Hills. Our genetics lab has this huge database across all the West on mountain lions. We're able to trace this individual all throughout its route, all the way to Connecticut, and actually assign it to the South Hills, this enormous migration. And again, when we quantify this as scientists, we're looking for exploratory movement corridors. And the last type of movement that I'm going to talk about throughout this brief talk is genetic movement, or phylogeographic movement. And that's this movement across space and time to set up these genetic patterns that we see today on the landscape. But I think these are also really important types of movement to consider. And we quantify that by using these phylogenetic trees, where branches that are close to each other, individuals close to each other that are connected by branches, are ones that have been more closer in recent evolutionary time than others. And so throughout my talk, I'll put, just to keep everybody straight so you know what type of movement I'm talking about, I'll use these little icons in the lower corner. When I'm talking about migration, like the elk migration, I'll use this little goose symbol. Territoriality, I'll use the bear symbol. Exploratory movement, you might see this set of binoculars, and then a lot of what I'm going to talk about is DNA stuff, because that's really what we've been spending a lot of time on the last few years, is trying to quantify substructure and movement and connectivity through the use of genetics. Okay, so the mesocarnivores, the mid-size or mesocarnivores. This is the group of 6 of them that we think about in this area, And no, they're not, I didn't put the same species twice. There are 2 species of marten, not subspecies, 2 species of marten that live in the Rocky Mountains. They have a line that runs diagonal from the northwest to the southeast. We have Martes americana and Martes crina, the western marten and American marten. So 2 distinct species, oftentimes get lumped as one. Canada lynx, fisher, wolverine, and fox. And when I first thought about this talk, I was going to give you information on all of them, and then I found out that I can't really do that in 30 minutes. I probably can't do that in an hour and 30 minutes. So we're going to focus on a few. We're going to focus on wolverine. We're going to focus, I'll give a little bit on lynx. So the bulk will be wolverine, a little bit on lynx. And then if we have time, and I just don't know how much I'm going to ramble, but if we have time, I'm going to tell you about this really cool new development that we have going in the lab that's going to help us understand both of these species. All right, so wolverine. All right, so the wolverine, a lot of you know about the wolverine, but so many people have so much misinformation about wolverine. And that misinformation starts back in 1552 even. If you go to like Olus Magnus, this is my favorite quote. It's one that Jeff Copeland pointed to me to a long time ago. And there's a wolverine in this picture. I don't know if I can get an arrow going here. All right, right in the middle, there's a wolverine there. It looks like he's being shot at by an archer. And kind of the first mention of wolverine is this misinformation. Wherefore this creature is most voracious, for he finds a carcass, he devours so much that his body by overmuch heat is stretched like a drum, and finding a straight passage between trees, he presseth between them, that he may rid his body of flatulence, and being thus emptied, he returns to the carcass. All right? This is our wolverine. So we hardly know anything about the natural history of wolverine, forget movements, right? But we learned so much more. Here's High Country News, that very reputable journal that we all read. Here's High Country News, that right in the opening start, this is in 1970s, right? 1979, like something from a nightmarish fairy tale, the wolverine is a threat to all creatures it meets. Another piece of maybe some misinformation. I don't know. So we've spent a long time in this myth of the wolverine without really knowing much. But I can say in the last 2 decades, through efforts from a lot of the folks here, from Montana Fish, Wildlife and Parks, Wyoming Fish and Game, Forest Service Research, and many, many other partners, we've learned a lot more about wolverine and a lot more about wolverine movement. So I'm going to talk to you about wolverine connectivity. And I'm going to start back at a very broad scale, and then we'll zoom in here. First thing I want to talk about is when we looked at wolverine, one of the first things we did is we said, let's look at wolverine connectivity across the world. It's a whole Arctic species. Let's look at their mitochondrial DNA. That's just one component of the DNA. And we gather those numbers are sample sizes. We and many other collaborators and folks that have worked in other labs tried to understand the pattern of wolverine across the entire geographic distribution. This is what I get to look at on a daily basis. This is a genetic code. If you read across in a row, that is one individual. And everywhere down is a certain spot in the genome. And you can see, if your eye catches somewhere in the middle there, you see, wow, there's a difference. For the most part, we have purple, and then there's a green. That one difference is what we call a haplotype. And those haplotypes are often reflective of certain geographic locations. And so what's kind of cool is if you find if everybody has a certain haplotype from one region and then someone else has a haplotype from another region, then you get a movement and you find this weird haplotype in the wrong region, you can start making inference to gene flow and movement. Okay, so we've looked at this in the Rockies, and here's another way of looking at the results from looking at the haplotype data. And yeah, I want to jump up there and point to things. But I'm not so good at sitting still, so I apologize if I start wandering that way. I'll try to stay near the microphone. But if we look at it, we have Scandinavia on the top, some samples that then came from Eurasia, China, Mongolia, and Russia, and then we get into the Rocky Mountains where we have all this genetic diversity. And I highlight four different circles there. Those are the haplotypes that we see most commonly in the lower 48 with this haplotype A, which is the one way on this left side here. That is about 93% of the samples that we've analyzed recently are this type. Now this is going to become important. I'm just setting the stage here for how this feeds into movement. We can build a little phylogenetic tree. It looks something like this. We see on that upper right by Alaska and Russia, we have one branch of the tree, and then everything else is this radiation this star radiation, meaning a recent kind of explosion of genetic variability in the areas in northern Canada and then throughout the Rocky Mountain chain. Okay, again, I'm just setting the stage here. The next thing we did when we started looking at Wolverine haplotypes and Wolverine genetics is we went and wanted to understand, where did California fit into this? This is before the recent guy that they call Buddy moved down there, that Wolverine that has been found in California, the one existing Wolverine in California. Before that individual showed up, the last Wolverine that was seen in California was 1923. But we wanted to understand, what's the relationship of California Wolverine to this whole group? So we went to museums all throughout the country. Actually, the biggest one was in Berkeley, where Arthur's at. I wish you were there now. It was kind of hard getting permits. Maybe you could have helped me out at the time. But okay, we got the permits. They were just fantastic to work with. And I got to sit. This is amazingly cool, right? I got to go to get samples that were taken by Joseph Grinnell, the famed early naturalist. They let me pull the turbinate bone. That's the lattice structure in the nose of a piece of a wolverine. We bring back that bone into the lab. I'm able to sit and take my notes right next to the desk that Joseph Grinnell was sitting at. It's just, here's his desk. It's for somebody that's a natural historian, you've gone to, it's like going to the Galapagos, or you've gone to Mecca. It's amazing. Or you go look at his notebooks. That's his notebook on the right. It's really exciting to be there and to feel like a piece of this history. We get these we get these bone samples. We bring them back and we get a few hair samples from a few of the pelts. We bring them back to the lab, and out of the 9 skulls, we were able to extract DNA out of 7 of those skulls. And that's my laboratory manager, Christy Pilgrim. And I have to mention, she's from right here. This is her hometown. I also have to mention her mom's back there. Wave, Judy. She's back there. Christy, everything I would do, I wouldn't be able to do anything without Christy. She's the most amazing lab technician. She makes magic happen in my lab. If it was just me, there'd be no talk. So she gets DNA out of 7 out of 9 skulls, and she gets 2 haplotypes, Cali1 and Cali2. Now what's cool is when we look at those, they don't come off this Rocky Mountain branch. They come off this other branch, suggesting that Wolverine colonized twice into North America. Once they came down and then colonized further south, then we had the second push, maybe the second wave of Wolverine that colonized to the north. Okay. You're like, great, that's great. Well, I'm in Yellowstone. Why are you telling me about this? Here's why. Another way to look at it, here's why. When we go and look at DNA, we go to a bigger, broader effort, and we say, let's go find all the Wolverine samples from all the museums that we can find. We go out there, and I don't expect you to read this, but this is samples from 1870 to 1915. And then into the 1920s, we have samples from Colorado, California, Idaho, Minnesota, Montana, North Dakota, Utah, Washington, and Wyoming. And when we look at these skulls, and this one from Wyoming, I should say, is from Yellowstone National Park. When we look at these skulls, we get a map that looks like this. And what's interesting about this map is if you think about the California type, this early colonization, that's in red and orange, and then we go back and look at these museum specimens, we see that there was some southern connectivity into Utah, into Colorado. Okay, we see these California types up into those areas. We also see some in southern Idaho. But what we see in the north, when we look at Montana, when we look at Wyoming, when we look at Colorado, we see these northern haplotypes. And so at some point, we know that these northern haplotypes moved down, were able to migrate down, and there was connectivity that went from northern Canada all the way down to Colorado. And we also know that we had some connectivity, not as far north as Wyoming, but up into this area, up into the Utah area. Okay. The interesting thing, from my perspective, is that haplotype that we saw historically, that was prevalent, that showed up sample after sample after sample, is now only found in a tiny place up in British Columbia in the Yukon. And that all other haplotypes that we see today weren't even really, we only saw a few of them. In fact, we only saw them over in the Cascades back in historic times. So what this is saying is that wolverines have gone through these pulse migrations and important movements through evolutionary time. And now when we look at the Cascades, we've gone from something that we now see in the Rockies to, you see that brown dot in the Cascade Mountains on Washington State, to something that's really different. So this haplotype C. So every individual that we see in the Rockies is this haplotype C, a very unique haplotype. Okay, so our mitochondrial DNA suggests that we've had movements over long terms, over long distances, and that Yellowstone was an important place of connectivity down to Colorado throughout evolutionary time. But let's turn to the nuclear DNA, where we can look at more real-time, kind of more recent movements. We went and collected with all our partners about 200 samples over 20 years, and we were able to look at nuclear DNA. We were able to test all these different ideas of how related are two individuals, any two individuals in our 210 sample database. Let's look at how related they are, and then let's come up with all these different hypotheses about how they could move. Did they move straight line distance? Did they move throughout spring snow, as it was in our case? And what we ultimately found out is that spring snow is an important driver of wolverine movement. From that nuclear DNA, we were able to say, what if we put a group of random dots in space, let them all connect to one another, and we find this map here suggesting that the greater Yellowstone ecosystem, if you want connectivity for wolverine into the greater Yellowstone ecosystem, it's the Centennial Ranges, it's these ranges that are to the west of us that are going to provide the important lifeline of gene flow for wolverine in contemporary times into the greater Yellowstone ecosystem. We also can see that if we take the map on the left, which is today, compared to the map on the right, which is pushing this forward two 30-year time steps with climate change, we can see that different connectivity, different pathways are going to become important in the future. So, today we may think about the connectivity coming from the west to the east, but in the future we may see more importance coming from the north if the climate change predictions are proven true. The other thing that became interesting is we made some predictions, and this was 2009, we made some predictions about how wolverine would move if they were ever to colonize the further southern parts of the range into Colorado. This red line, you can see, we said the Wind River Range is going to be hugely important for connectivity, moving down into the San Juan Range. You'd follow that red line. If we were going to use our gene flow data to make predictions, we'd think that this is the way that wolverine would be moving into Colorado. Fortunately, Bob Inman had a whole study with radiotelemetry animals throughout just after that time period, 2009 and onward. And what's cool is that one of his individuals, one of his wolverine, actually took a path down there after we made this prediction that almost followed precisely what we would have predicted with our gene flow maps of how this individual should have moved. So we're going from genes to predictions of corridors and the importance of the Yellowstone area for not just individuals coming into the area, but putting some propagular pressure to let individuals go out of the area to colonize places like Colorado that currently don't have wolverine. Okay, that's how we're using genetics to make some inference into the importance of connectivity to the Yellowstone area. I briefly want to talk about lynx. Lynx, we can talk about the genetic data. This is some early work that we did. Lynx are one of those species that really can move. And we think of lynx as a tide pool model. They build up to huge population numbers in the central part of Canada and the United States. And then the hare numbers crash. Lynx try to disperse. When they disperse, it's like the tide comes in and fills up all the tide pools. And then as the lynx numbers dry up and as the hare numbers are at the low cycle, we have a few areas the tide pools that fill up, like the southern U.S. That's kind of our best understanding. When we first looked at genes in lynx, we found connectivity up and down the Rockies. We found almost complete panmixia, almost complete mixing of genes. You couldn't differentiate a lynx from one part of the range to the other. And that's really interesting if you look at genetic distance on one axis and distance on the other. Let's look at bighorn sheep. Anytime you move slightly one area to the next, you would see huge genetic distances, suggesting you don't have much gene flow. Not surprising. But something like lynx, if you look at the straight line on the bottom, any physical distance will lead to genetic distance. You get no genetic distance, suggesting a lot of movements of genes over time and space. And so we had this initial idea that lynx were moving fairly freely using our genetic information over time up and down these ecosystems. Well, what about the telemetry data? And here I'm trying to keep you straight by putting the binoculars there. This is from the Squires and Oakleaf paper in 2005. He had a lynx that had a home range that's down here south of the park or south of the GYE. That individual made 3 movements that went, first it made a small movement, you can just see where the black line is, moves up to the northwest, and then for 2 years in a row made movements through the park, exploratory movements, and then back down towards that home range. So lynx have been using, at least in an exploratory sense, the Yellowstone ecosystem. What's given us a new way of looking at this are all the reintroduction efforts that occurred in Colorado. And the next group of slides are from Jake Ivan from Colorado Parks and Wildlife. He sent me these last week. And these are individuals that were released in Colorado who made exploratory movements either up through the Wyoming range or up through the Wind River range. And again, lynx are using this habitat. Really, they're coming up there, they're exploring, and then they seem to be going back down to either to Colorado or back down to southern parts of Wyoming. And we see this again. This is BC04. This is BC06. Here's one, a QU04 individual who went up into the Yellowstone ecosystem and stayed there for a period of time. You can see a cluster of samples where it stayed for a while. And we see this over and over again. About most of the lynx in Colorado stayed in Colorado, but a fair number of them came up through the GYE, and it became an important place of connectivity. Now, what's also cool is at the same time, we had a lot of groups, some Forest Service folks, some nonprofit groups that were out there backtracking putative lynx tracks to pick up hair samples or fecal samples and sending them to our lab. And we were able to look at those to, 1, confirm that yes, they were lynx, and 2, to be able to assign those to one place or another. And if we look at this group here, each of those individuals, there's 3 clusters of individuals, each of those assigned to a known Colorado lynx. So we have people on the ground picking up hair samples, seeing the tracks of these lynx as they move through the ecosystem. And that one I just circled turns out to be this one here. And if I had a pointer, you can see that cluster in the middle. That was at that time. The sample was picked up at the time when that individual was known to be there with a radio collar, which I think is a pretty cool observation. The sample on the top here, this sample is really cool because when I look at that sample, it matches none of the Colorado samples. It in fact assigns, when I do a genetic assignment test, a kind of a forensic test we use, it assigns to the northern population of lynx. It's got an 89% assignment rate to that northern population of lynx. That suggests that Yellowstone ecosystem is acting as an important area of mixing these southern lynx coming up from Colorado, and again, these northern lynx coming down from Montana and the Sealy Lake population and populations in the Purcells in northern Montana. Okay, so I got a couple more minutes. I promise I'll try to finish this up in a few minutes. So right now, what we have is a situation where we have the greater Yellowstone area not being settled by lynx, but where lynx are coming up and through the area using a lot of exploratory movements. A couple of times they're using it as almost like Arthur's stopover habitats. They'll stay there for a while and then move on. We have some lynx from the north coming down, but we also now have no more collars being put out in Colorado, or at least a much reduced effort to collar lynx. So how are we going to figure out how many lynx, or are lynx still using the greater Yellowstone ecosystem? And in the next 2 or 3 minutes, I just want to show you quickly a new technology that we've developed, and it's a paper that's currently in progress. We should have it out sometime soon. That's a new approach that I think is going to help us understand lynx in these ecosystems, so rare species and rare ecosystems. So we know we could try to follow tracks, and we could try to get to follow a track of a putative lynx, and about every kilometer and a half, we get a useful DNA sample. So that's one thing that we could do. What we've started to do recently is our genetics lab has really gotten into this environmental DNA technology. It's blowing up my lab, in fact. If anyone has a good business management sense, come see me. We guess about 20,000 samples that we've run in the last 3 years using this technology for all suite of species, all manner of species. And what it is, you scoop water. In your cup of water, you're finding the one cell that determines if that invasive species or if that endangered species is in that body of water. I mean, it's just mind-blowing that you can do that, right? So we're able to do that for fish species after fish species. Now we're doing it for a whole suite of terrestrial species. But one of these crazy afternoons, we had one of these meetings where we decided, hey, you know what? Snow is nothing different than just water. What if we scoop snow, bring it back to the lab, run it through our environmental DNA protocols, can we detect a species? First thing we did, thanks to Scott Jackson from the National Forest System, gave us a little exploratory money to try this out. We could take old snow tracks, we could take new snow tracks. We built a marker for lynx with a little bit of seed money he gave us. And we're able to go out there, and if you look on the left, it's number of tracks. It says 554, 86, 11, 262, small number of tracks you're scooping. Track age, fresh tracks or really old tracks that may have been lynx or may not have been lynx. We tested them with our lynx marker, and we had complete success. So now we're at the place where we don't have to wait till that fresh snow comes about to scoop snow to understand, is that a lynx? All of you guys know this, it's so limiting. You have to wait 24 hours after snow before I go out there to see if I can find the right track to make the perfect print. Well, now you don't need the perfect print. You need kind of a sort of idea it might be a lynx. Go scoop some snow and bring it back to us. We now can determine if it's a lynx. We didn't stop there because it was just too exciting, right? We're like, okay, let's get some markers. And we partnered with MPG Ranch, and they gave us some funding to build a genetic tool for wolverine and another tool for fishers. And so the same thing, old, fresh, and we look on the far right, we see fisher, lynx, wolverine. We ran those in the lab after spending a few months developing the technique, and we had success there as well. So this is this is getting exciting, right? Now we have 3 species we can test for. We want to build 10 more. I'm trying to keep all my technicians under control, not letting them just spend their weekends building markers for rare species, right? But then they go even further, and we were working in the Beaverhead Deer Lodge, and we find this picture, lynx, bobcat, I don't know. I mean, the tail, which one? I mean, they both have that black little tail. Most people say lynx, some people say bobcats. We're like, well, okay, that picture was taken before we had this technology developed back in November. What if we dug a pit back to where the snow level should be in November? Could we actually get that DNA? It should be frozen. I mean, what are we going to do in the lab? We're going to put it in the freezer right when it comes in, right? So we try this. We collect 3 samples, one from a sample from November, one from a sample in January, and then one at the base of the bait tree. We run it. The November sample, we get positive for lynx, not positive for any of the other species that we test for. The one at the base of the tree, which we didn't know there was a lynx there, that's positive for lynx. We had a failure in the middle, but this is science. Don't expect perfection. Okay, so yes, 2 of our samples are lynx. Here's the thing. That April 13th sample, I mean, that one that you see at the bottom, what we collected at the base of the tree, it turns out that a week later, we got a photograph of a lynx visiting that site. So we didn't even know the lynx was there recently, but apparently had been back in the area one more time, and we had picked it up with that sampling right at the base of the tree. So now we have a technology that we can go scoop snow, and even in places where we don't have radio collars on, we can start to see, so like the greater Yellowstone ecosystem, we can keep monitoring this ecosystem to see how important it is for movement and connectivity into, from Colorado or north from Montana, and acting as a connector between 2 populations of lynx. So, the conclusions. There's a lot of conclusions. I think the first thing is that I want you guys to remember mesocarnivores don't migrate, okay? I don't want to show that Wolverine video one more time. They don't migrate, but they do move. They move across evolutionary time. They move in recent times. We can understand those movement paths, those places of connectivity, because if you don't have connectivity, you create little isolated zoos on the landscape that are very expensive to manage. You have to keep moving individuals in. You keep having to take a very heavy-handed management approach. We don't want isolation. So mesocarnivores will move even if they're not migrating. Second, greater Yellowstone ecosystem has been very important for long-term movement, as we saw with the ancient DNA. results for wolverine. But it's also been really important for movement in the short term, as we've seen with lynx moving up and down and with some of the wolverine examples. A very important critical connectivity step for movement. And finally, we have some new technologies that I'll leave to you folks to apply in a creative way to help us all understand how these individuals are using the ecosystem. We have a new tool to help us conduct these surveys and to understand lynx, wolverine, and fisher movement. And with that, before I take any questions, I just need to thank, my name's up there in the front, but I do probably the least amount of work of all the technicians, all the fishing game, Montana Fish, Wildlife, and Parks, all our Wyoming Fish and Game, our fantastic partners from the National Forest system all the way through the Park Service. These are the people that are really doing the amazing work that I'm fortunate to be able to come up here and represent. So thank you very much.
Mike, I've got one for you. On that snow sampling that you guys did on the BD, did that come down to individual level or is it just species level?
You always want more, Kerry, don't you? No, we're just at species level at this point. I don't know if we'll get to individual level. I think we could get to haplotype level, meaning we could understand something about if we had a wolverine show up, we could understand which population it came from, but that would take a little more. We're not close to that yet. That's kind of the science fiction next step.
Thank you. I was just wondering, at the beginning of your discussion, you showed the graph of abundance, of showing wolves, bears, and then the abundance going down. What do you mean by abundance, and what does that mean in terms of numbers? Is it in geographical areas? Is it population across the board, or what?
No, it's a great question. I mean, abundance can be, there's a lot of, first of all, we don't know very much about abundance of those rare carnivores. You know, the wolves and bears, honestly, I went to the reports, the recent federal reports, I tallied up the numbers in the Greater Yellowstone ecosystem and also up through the Northern Rockies to the Canadian border, and I put those, that's how I got those two numbers. For fisher, it's literally, I've sat around with folks from the different state agencies and said, okay, at best, what do we think we have in this mountain range or that mountain range? How many individuals could we have here if we took our best estimate possible? And with fishers, like if you sit there, I've heard people say as many as 600, but then when you sit down with them and you try to add it up, 350 is the best you can get. Wolverines, again, we don't really know, but we know what Washington State, I recently did that with the lead biologist out of Washington State, most they can think about is maybe 35 to 50. California has 1. Utah, it looks like, has 1. So now we're at 35 plus 1 plus 1. Montana, this area here, again, very hard to quantify. People have tried to do it by saying, how many home ranges can we fit in here? And then you can get up to an estimate of 500, but that's super optimistic that everything's being used. We just don't have good abundance estimates of these carnivores, these rare carnivores. And lynx is probably the worst. So we just know they're incredibly rare and at a much lower density than the two species that we often think about as being rare in the ecosystem. Yeah, thanks.
When you showed the lynx movements out of Colorado, are male and female animals moving like that?
Yeah, that's the amazing part to me, is that we did see males and females. A lot of those individuals were, I think more of them were females than males. The one from the north coming down was a male that came down that assigns to the northern population. Yeah, it surprises me because obviously males move long distance, are known for mammals to be moving the long distance exploratory movements. But one important thing is we're not seeing a lot of those individuals move and then settle. It's move, explore, and then oftentimes return.
You're going to get to hear someone who knows some stuff about ungulates show off how little he knows about the species you're talking about. But I just wonder, so it's fascinating to read some of the historical accounts of journeys through the ecosystem. Osborne Russell, the trapper, is one of the few members of that kind of mountain man clan who wrote, and there are these accounts of these huge historical densities of bighorn sheep up at the high elevations throughout the ecosystem, and you see some of these other species I talked about being less important in those accounts. And so I wonder if those of you who think about wolverines knowing the importance of those high elevation mortality patterns and carcasses, is that a part of how you think about the rarity of wolverines in this landscape? Is this a dumb question?
No, it's a great question. It's a great question. Like, rarity, when I'm talking about rarity, I'm talking about just overall abundance. And you're saying, well, how does it compare to historic times? Were these things ever common here? And honestly, we have such poor information on historical records for a lot of these species. I've done a pretty extensive look at fissures, for example, and trying to understand fissure records anywhere in the Rockies. They're just completely absent. I mean, just completely absent from the record. But when you look at genetic signals and genetic patterns, you can see that there's areas of abundance that had to be somewhat common. You can't get the type of genetic diversity that you find in some of these areas if it was a recent colonization. You'd expect much lower levels of genetic variability. So we know that we believe that they were always there, but why they weren't showing up in the trapping records, it's unclear. Were they called something else? Were they not differentiated from marten, so on and so forth. Wolverines, it's really hard. You're right. I mean, Wolverine records are, the records are all over the place, and you see something where you might see a record from a location, but it's not clear if it was actually collected in that location or if that's where it was sold. It's just I haven't seen. Our historical understanding of these species is really poor, even in going through the best of journals. So, yeah, I wish I had a better answer than that, but I appreciate the question. Yeah, how rare are they now compared to what they have been? And again, looking at genetic types, looking at types, the historical movement through California, through the Rockies, we see the abundance of genetic variability up north, suggesting that they've always been more common up there. And it's always been a little bit scant down here. So my guess is they've always been somewhat rare down in this part of the world. And they've probably, because we've seen changes in haplotypes, they've probably also blinked out. Small population blink out, get recolonized from the north, and stay there for a while. So it's a good question. I just wish we had a better answer.
Mike?
Okay.
I just wondered why they're exploring and then coming back.
Me too.
Okay.
Yeah, I don't know. Honestly, it's the same with that lion that went out to Connecticut. Oftentimes it's juveniles. Oftentimes you find this more often than not in places that have propagule pressure, meaning that the landscape's saturated. I think they make the mental calculus. The odds of me surviving, finding a mate, and staying here are pretty low. I'm going to do the mental calculus. I'll bet you there's something better over the horizon. It's why we found the New World. So let's go to Connecticut. Let's go see. Let's just keep going until I find something. I honestly, it might be that simple. We don't know. I can't get into the psychology of a lynx deciding to move, but oftentimes we see that when areas, with the case of the lions, for instance, we know that the South Hills was getting pretty dense with lions, right? So there was some propagule pressure to move some of those juveniles who won't compete well out. That's the common textbook wisdom. It's a great question, though. All right. Thank you very much.